IKEMA is a phase 3 randomized prospective study that did compare for patients with relapsed multiple myeloma, carfilzomib and dexamethasone, one of the standard of care, versus carfilzomib-dexamethasone plus Isatuximab that is targeting CD38 antibody. And we have presented during the IMS meeting the overall survival analysis of this study. The study was already presented in different meetings, have been published already, and is the basis of the approval of carfil-dex plus Isatuximab based on a very important progression-free survival benefit with the triplet combination. And the PFS, the final PFS analysis was already reported with a median PFS of 35.6 months for the ISA-KD arm versus 19.2 for KD. And here we have presented the overall survival data with a median follow-up of 56 months, and the median OS with KD is 50 months, and the median is not reached with ISA-KD. And the projected the median OS estimate for the ISA-KD arm is 63 months, so there is probably 1 year difference in median OS in favor of the triplet. So this is not statistically significant. There is only a trend for a better overall survival, but that's a very important finding for sure. And we already reported the time to next treatment in favor of ISA-KD, the PFS2 as well, the time from randomization to PD on subsequent therapy or death, that is also in favor of the triplet combination. And we don't have any new safety signals. So definitely this triplet KD Isatuximab is a very interesting one for relapsed myeloma patients. So during the meeting, we have heard for the first time about the results of the CANOVA study, a phase 3 trial comparing in patients with relapsed myeloma with 11-14 positivity, venetoclax dex versus pomalidomide and dexamethasone. 263 patients were randomized. The dose of venetoclax was 800 milligram daily, and the primary endpoint was PFS. The patient characteristics were well balanced in the two groups of patients, and the primary endpoint PFS was longer with venetoclax dex versus pomdex, 9.9 months median versus 5.8, but disappointingly did not meet statistical significance with a hazard ratio of 0.82 and a p-value of 0.2. So numerically longer, but not statistically longer, and that's disappointing for sure. When we are looking at time to next treatment, it was also longer with Vendex versus Pomdex, 21.2 in median versus 8.3. The responses were higher with venetoclax dex, 62% of response overall versus 35%. And the MRD negativity rate was also higher at 10 to the minus 5, 8% versus 0%. And the OS was also numerically longer with Pomdex, with venetoclax dex versus Pomdex, 32.4 months versus 24.5. I would say disappointingly, because the study was a pivotal study for the approval of venetoclax and unfortunately did not meet the primary endpoint. We have heard also about other results with venetoclax in combination with carfilzomib-dex versus carfil-dex alone. That's a phase 2 study in patients with 11-14 positive relapsed myeloma. And the dose of carfil was a weekly dose at 70 in combination with dexamethasone, 40 milligrams weekly. and two different doses of venetoclax were tested, 400 and 800. And that's very interesting to see that the responses were much higher with KD venetoclax, 82% versus 63% with KD alone. And when comparing the two doses of venetoclax, 400 or 800, there is really no difference in terms of overall survival. That's roughly 80%. And the PFS with venetoclax carfildex was much longer as compared with KD alone, median 32 versus 14. So that's really amazing. In fact, there is no difference in terms of overall survival, but the addition of venetoclax to KD is really improving the outcome of patients. And from a safety point of view, obviously with venetoclax, more GI toxicity, the neutropenia rate, the thrombocytopenia rate was also higher, and a higher rate of infection versus KD alone. So these are only phase 2 data that will not support venetoclax in combination with KD for an approval in front of the authority. We have heard about two interesting studies looking at high-risk, newly diagnosed multiple myeloma, one from the German group. The name of the study is the CONCEPT study, and patients were treated with a very aggressive treatment with Isatuximab KRD, followed by autologous stem cell transplantation, Isatuximab KRD consolidation, and maintenance with ISA-KR for transplant-eligible patients. So 99 patients eligible for transplantation were treated, and the MRD negativity rate at the end of consolidation before maintenance is indeed very, very high, and the progression-free survival is also very good, 84% at 1 year, 77% at 2 years, and 68% at 3 years for PFS. And for overall survival, we also have very, very good data. The 2-year probability of survival is 84% for this subgroup of high-risk disease. So interesting data for sure, showing that we need to intensify the treatment for patients with high-risk disease. And our German colleagues looked at PFS by high-risk features, and they showed that those patients with elevated LDH, more than 2 cytogenetic abnormalities or 17p deletion, have a worse outcome as compared with others. So this is quite interesting. Another study coming from France with the French IFM group, looking also at a phase 2 study in patients with high-risk disease. And high risk was the definition of the International Myeloma Working Group by Cytogenetics, high-risk FISH, either 414, 17p, or 1416. And we have proposed a quadruplet induction with KRD daratumumab, a tandem stem cell transplantation with 4 cycles of consolidation with KRD dara in between the 2 intensive treatments, and a maintenance with 2 years of len and daratumumab. 50 patients were enrolled, all of them with the high-risk disease, and the response rates and MRD negativity rates are very, very high. In fact, with 60% MRD negativity rate after induction, 85% after stem cell transplantation. So that's very, very good results indeed. And the progression-free survival is also incredibly high. The 2-year PFS, 86%, and the 2-year overall survival, 94%. So those data are suggesting that maybe now we can treat patients with high risk or standard risk differently with a very aggressive strategy to have a good outcome. But the treatment is very long, indeed, including tandem high dose, including a long maintenance. And the last presentation, maybe that it was interesting during this meeting. Well, plenty of them were good for sure, but I have selected the CARTITUDE-4 trial. That's a randomized study, a phase 3 trial looking at ciltacabtagene, a CAR T cell, versus two standard of care, either DARA POMDEX or POMVD, and the prospective comparison already reported showed that the median PFS with the standard regimen was 12 months, approximately 1 year for those patients with relapsed myeloma, and versus a median that was not reached and a hazard ratio of 0.26. So very good results in favor of the CAR T cilta-cel. So now the goal of the study was to look at some correlative analysis of cilta-cel to try to identify those patients that will have a better outcome with the CAR T. And we saw that there is a preferential CAR T CD8 T cell expansion that is correlated with a greater clinical activity, and also that there is, for those patients with a better outcome, an enrichment with T cells possessing memory phenotypes. So those informations are important because we would like to understand better how the CAR T are some expansion of CAR Ts, some subgroups, some subset of CAR Ts are associated with a better outcome. So many new data during this meeting. It is difficult to summarize the whole meeting with few take-home messages, but more and more, we for sure are going to use immunotherapy in the future. We have a lot of data with CAR T and bispecific antibodies for very advanced patients, but those therapies are moving now at earlier lines, and that's very important findings. We have now probably a new definition of patients with high-risk disease, and the outcome of our patients and the survival for sure is highly increasing.